Hi guys, welcome back to my channel. I'm so happy to see you all here. So after a lot of you requesting a tutorial on trajectory analysis using Monocle3, I finally decided to create a video. So this video is going to be about constructing single cell trajectories using Monocle3. So for today's video, we'll be covering a lot of ground, starting with what is trajectory analysis, followed by when to perform this analysis, after which we will talk about which trajectory inference method to choose, and lastly, we'll talk about how to perform this analysis. In that section, we'll also look at the workflow steps, and we'll also talk about some of the objects that Monocle3 uses. We'll basically talk about the structure of that object, and lastly, I will demonstrate how to perform this analysis. When studying dynamic cellular processes like cell differentiation or cellular response to a certain stimulus, cells transition from one functional state to another. So when these cells move between states, there is a lot happening at the transcription level, meaning that there are certain sets of genes that are activated during the earlier states, and there are certain genes that are activated during the later states. Instead of purifying cells into discrete states experimentally, which is also challenging to do at times, what Monocle does is that it uses an algorithm to learn the sequence of gene expression changes each cell must go through as a part of dynamic biological process. So basically it learns based on the gene expression patterns whether the cell belongs to an earlier state, an intermediate state, or at a later state. So once it has learned a trajectory of gene expression changes, Monocle can essentially place each cell at its appropriate position in the trajectory. And if there are multiple outcomes for the process, Monocle will construct a branch trajectory, where each branch would correspond to a cellular decision. And Monocle provides us with powerful tools to identify genes that are affected by these decisions, and also the genes that are involved in making these decisions. We measure the progress of an individual cell through a process using a numeric value which has arbitrary units, and we call it pseudotime. So pseudotime essentially measures how far a particular cell is within a dynamic process. So cells in the earlier state would have smaller pseudotime, and cells in the later state or intermediate or later state will have like higher pseudotime. So by ordering the cells according to pseudotime, it becomes possible to define the different transition states through which a cell progresses during its dynamic process. Now talking about when to perform trajectory analysis. So if the goal is to study biological processes like mechanisms that play a role in development of resistance to a therapy, or studying immune responses or rare cell populations, or understanding cellular heterogeneity in your data, or to even understand clonal evolution and the role of microenvironment in cancer, performing trajectory analysis makes sense. Before running trajectory analysis, it is important that you check several assumptions, like the process that you're trying to study is dynamic process. You have prior knowledge and or evidence that a trajectory exists in your data. You're ensuring that appropriate cells are sampled and the data is sampled to sufficient depth so that you're ensuring the presence of transition states in your data. So if you're examining a stem cell differentiation data set, then trajectory analysis seems to be the logical thing to do. But if you see your data being clustered as distinct clusters and the cell types are not known to be associated by any known biological process, then trajectory analysis may not be appropriate. So looking at t-SNE and UMAP plots can also be sometimes useful to decide whether a trajectory analysis should be performed. Now talking about trajectory inference methods, there were 59 trajectory inference methods according to a Nature Biotechnology article that was published in 2019. I'm sure there are many more now. So the question arises as to which trajectory inference method should one use for the data. And the choice of the method would depend on various factors like whether you expect disconnected trajectories in your data, do you have prior knowledge or do you require prior knowledge to run the method. In addition to that, do you expect any particular topology in the data, whether the tool is user friendly, whether it's compatible with the system that you're using, as well as with the other packages that you use to pre-process your data, as well as the performance of the tool. As for the performance of the method, it is most heavily dependent on the topology of the trajectory, and it's also heavily influenced by the user's existing knowledge about the expected topology of the data. So each method might have its own strengths and weakness, and also has its own set of assumptions. Like certain trajectory analysis methods would assume that all the cells that are present in the data belongs to the trajectory. So this might call for rigorous pre-processing steps to make sure that the noise or the outlier cells are not used to construct the trajectory. So Dinverse is a collection of R packages which provides various functions to perform trajectory analysis. So it not only allows us to perform the trajectory analysis, but it also provides us with an R Shiny app that allows user to decide which method is more appropriate for their own data set. So it asks a bunch of questions and suggests you the methods which are most appropriate to be used for your data. In addition to that, it also tells you the amount of time and memory that will be required to process and build a trajectory for your dataset. So finally, talking about how to perform trajectory analysis, and before I jump on to the demonstration, I want to talk about the workflow steps. So these are the workflow steps taken from the Monocle3 documentation. So here we start with the pre-processing of the data, which involves normalizing and removing batch effects. Then we move on to nonlinear dimensionality reduction. We cluster the cells. We compare the clusters to identify top markers, which helps us to identify and annotate the cells, identify the cell types. And lastly, we perform the trajectory analysis. So trajectory analysis is usually performed after clustering your cells and annotating your data set. So Monocle has functions to process your data, to perform non-linear dimensionality reduction, to cluster your cells, to identify top markers, and to perform trajectory analysis. But today we are not going to use all the cluster Monocle3 functions. We are going to use Seurat. So the data we are using today is already annotated. So to perform pre-processing, non-linear dimensionality reduction, and clustering, we will be using Seurat, and then we will move on to Monocle3 to perform trajectory analysis. An important thing to know before we jump onto the demonstration is that Monocle3 requires data to be in the form of a cell dataset class. Now, cell dataset class is derived from the single cell experiment class. Basically, these objects are slightly different from the Seurat objects. The way that the data is organized in these objects are a little different. A good thing to know about Monocle3 is that it takes the count matrix produced by Cell Ranger pipeline, which is often used to analyze experiments from 10x genomics instruments. So basically, this cell dataset class requires three input files. The first is the expression matrix, which is essentially a count matrix where rows are genes and the columns are cells. It requires a cell metadata, which is essentially a data frame where rows are cells and the columns are attributes like cell types, culture conditions, or the day captured. And third file that it requires is the gene metadata, which has information about the gene, where the rows is again a data frame where rows are genes and the columns are attributes about the genes, like just GC content of the gene and more information about the gene basically. So if this might not make sense, it will start making sense once I start with the demonstration, and I can practically show you what each of these files look like and basically how the data is organized in the cell dataset class. For the demonstration today, the data we will be using is a transcriptomic landscape of human blood cells. So these are essentially 7,551 human blood cells covering 32 cell types from 21 healthy donors. But for the sake of demonstration today, we will not be using the entire data set. We are just choosing a group of cells, a group of B cells and its progenitors, which are around 1400 cells. So we will be building a trajectory using these group of cells. So the goal of today's analysis is to construct a trajectory, to order cells in pseudotime, and finally find the genes that change expression as cells progress along a trajectory. So for the demonstration today, we will be using Monocle3 Seurat, Seurat wrappers, and tidyverse to perform trajectory analysis. So let's switch screens to RStudio to get started. So before jumping onto RStudio, let us first get the data that we want to use today. So the data we require is deposited on this website, and I will be adding the link to this website in the description section below. So let us go to the download section here, and we will be downloading the gene expression matrix, the marker information, and the cell metadata. Since I've already downloaded these files, I will not be doing it again, but essentially these are the three files we will be requiring for today's demo. Once we have finished downloading the data, let us start by loading the libraries first and reading in the data. So we'll load the libraries and reading the data. So I have downloaded the data already, so I'll start reading my files. Once I read in the files, I will show you the contents of these files. Now that we've finished downloading the data, let us take a look at the contents of these data frames. But before we do that, recall that from the presentation, I mentioned that Monocle3 requires cell dataset object. And to build a cell dataset object, you need a cell metadata, you need gene metadata, and you need expression matrix. So essentially, these three files correspond to each of that. So we'll take a look at the contents of these data frames. So looking at the markers data frame, here we have information about the gene. So this is the gene metadata. And now let us take a look at the metadata here. So here you can see that we have cell IDs and we have more information about each of these cells and also what cluster it belongs to. And since this is an annotated data frame, we also have the annotations here. So this makes this data frame cell metadata. And lastly, let us take a look at the expression data frame. So we can see that here we have cells as row IDs and genes as column names, which is actually the transpose matrix. This is the expression matrix, but it's transformed. We need the rows as genes and columns as cell IDs. So essentially, this is our expression matrix. So if you were to build a cell dataset object, you can use these three data frames to build a cell dataset object and then proceed through the Monocle workflow to pre-process your data, to cluster your data, and followed by that you can perform the trajectory analysis. But today we are not doing that. We will be going through the Seurat route first. So I will be using the expression matrix and the cell metadata to generate a Seurat object, and then we will be performing the pre-processing steps in Seurat and then transition to Monocle. So let me show you how we do that. So the first thing we are going to do is we are going to create a Seurat object. And for that, we need to first transpose our expression matrix because we need the genes as rows and the cell IDs as the columns. So we are going to transpose the expression matrix. And let us take a look at the transposed matrix. Now we have the rows as gene IDs and the columns as cell IDs. So this is exactly what we wanted. In the next step, let us create a Seurat object. We will use the function create Seurat object. The counts will be transposed expression matrix, and let us assign to a variable. Now that we have created a Seurat object, let us take a look at the metadata for this Seurat object. So essentially for the metadata, we can see that it has the number of counts and number of features for each of the cell IDs. So we also need to add additional metadata here, which includes the annotation and a bunch of information that we have in the cell metadata. So let us add the cell metadata information to this metadata. So we can merge the Seurat objects metadata with our cell metadata. And we would essentially want to merge it by a column. So in the Seurat's metadata, we will be using the row names. So by X would be row names and by Y would be the cell IDs. And let us assign it back to Seurat object. Now that it has merged, let us take a look at the Seurat's metadata again. Now we can see that in addition to the number of counts and the number of features, we also have additional information like the individual the cell is from, the population, and also cluster annotations. One step we need to do is that we need to convert this column into row names again. So let us do that. followed by that, let us calculate the percentage of reads that map to mitochondrial genes. So we do that by %feature set. The object is Seurat object. Pattern of the genes for mitochondria start with empty, and let us assign it back to Seurat object and create a new variable called mito%. And after that, we will be performing the filtering of our Seurat object. So basically, we will be filtering out the low quality cells. So you know the drill here. So we do this routinely for all our single cell analysis. And essentially, we will filter out all the cells having less than 800 reads. Also, we will be filtering out all the cells having less than 500 genes, and mito% having greater than 10% of mitochondrial, having greater than 10% of the reads mapping to mitochondrial genes. And let me change the name of the object to filtered so that we can distinguish between the unfiltered and the filtered Seurat object. So after performing filtration, what I essentially want to do is I want to subset my Seurat object for it to only contain B cells, because we are only going to use B cells to perform further analysis. So let us first take a look at some of the annotations for the cell. So I'm just going to do a unique on my filtered object, and in the metadata I'm just going to look at the population. So all of our cells belong to either of these populations, which is stem cell progenitor cell, T cells, monocytes, NK cells, erythrocytes, B cells, or neutrophils. And what we're interested in, all the cells belonging to the population B cells. So I'm just going to subset my Seurat to only contain the B cells, and then I'm going to perform the pre-processing and dimensionality reductions and all those processing steps on only the subset of my cells. Let us start by subsetting my Seurat objects first. So the idents for my Seurat object filtered is some other identity, and I want to change the identity to population. So because I want all my cells to either be labeled according to the population, according to what group it belongs to. So I'm going to use and change the idents to population. And let us run the idents again, and now you can see that all the cells either belong to one of these cells. So that's exactly what I want, because that will help me to subset my dataset, or rather Seurat object. So we can subset it using idents, and I want to subset all the cells belonging to this population B, and now I'm going to label it as B Seurat, and we run this. So now that I've subsetted this, let us just take a look at B Seurat object, and now I can see that I have these many genes and over 1400 cells. So these are exactly the number of cells I wanted, because these are all the cells that belong to the B cell population. Let us also take a look at what are the types of cells that are present in our data. Let us go into further annotations. So we can take a look at redefined cluster. And when I run this, we can see that we have the pre-B, pre-regulatory B cells, immature B cells, naive B cells. So these are all the population of the cells we have retained in our data. Here we can see that the B cells among the B cell populations are in various stages, right from the progenitor B cells to more mature B cells. So this would be an ideal data set to perform trajectory analysis, because we have not only the start and the end state, but we also have the intermediate stages of a cell, which will help us construct a trajectory and also take a look at genes that change along the trajectory. After subsetting our Seurat object, let us now perform the pre-processing and processing of the data using Seurat. So we start by, we perform normalization, we scale the data, we perform linear dimensionality reduction, we find clusters, and we also perform the non-linear dimensionality reduction. We do all of that in Seurat. So we've done this previously, so I'm just going to utilize those functions here. So we first normalize the data. Followed by normalization, we find variable features. After finding variable features, we scale the data. After scaling the data, we run linear dimensionality reduction. After running the PCA, next we will find neighbors in order to cluster our data. So we will be using first 30 dimensions, and followed by finding neighbors, we then find clusters using resolution 0.9. I want to point out that trajectory analysis is heavily dependent on the topology of the data, and the topology is determined by the clustering. So it is very important that you cluster your data using the optimal resolution, making sure that each cell type groups together as distinct clusters, and similar groups of cells do not get clubbed together in one cluster. Because this might be important, as there might be transition states of cells which could be easily grouped together if you're using a lower resolution. So make sure you're using the optimal resolution to cluster your data. And after finding the clusters, we run UMAP using first 30 dimensions, and we will be setting the n neighbors to 50 and assigning it back to the Seurat object. Setting n neighbors to 50 gives me a nicer grouping overall. So hence I set the n neighbors as 50. And now let us visualize our data. So we can use reduction UMAP, and we will be grouping the cells here by redefined cluster, and we want the label true, and let us assign this to a variable called a1. Let's create another plot where we want to visualize the clusters. So we will group it by Seurat clusters and label this true and assign it to another variable. So now we can visualize these plots side by side. Zooming in on the plot here. So looking at these plots here, I think my clustering resolution has done a fair job in terms of separating or grouping the major cell types into distinct clusters. Although I do see some amount of overlaps between some cell types, but I think some major cell types like Pro-B, cycling pre-B, memory, plasma cells are grouped into separate clusters. So I recommend you to play around with resolutions when you're clustering your data to ensure that you do not over cluster or under cluster your data, but make sure that the major cell types in your data do group together into distinct clusters. So I think my clustering resolution has done a fair job, although it's not ideal because I do see some clusters here which are not technically distinct clusters. But among the other resolutions, I think this one has worked the best so far. So I'm going to go ahead with this one. If you're wondering how to find the optimal resolution, it's an iterative process. So you run your clustering step with various resolutions, and then you visualize your data as a UMAP. You can also take a look at the feature plot, looking at the expression of certain markers that you know are expressed in certain cell types. So that can also help you determine whether these cells are separated out in clusters, like distinct clusters. And until you do not see distinct clusters for distinct cell types, you repeat the process with different resolutions, and finally end up with the one that provides you with the optimal separation of cells according to their cell types into distinct clusters. So quick recap on all the steps that we have performed so far. we read in the data into a Seurat object, and we performed the pre-processing steps which involved filtering the low quality cells, normalizing and scaling the data. We performed linear dimensionality reduction, non-linear dimensionality reduction, and we also clustered our cells. And we did not have to annotate our data because our data is already annotated. So we have performed all these steps in Seurat. Now the only step that is remaining is to build a trajectory, and for that we need to switch to Monocle3. Now, since Monocle requires an object of ScaleData class, we need to convert the Seurat object into an object of ScaleData class. So Seurat wrappers has a function called as ScaleData, which can convert the Seurat object into an object of ScaleData class. So after running that, when we just take a look at the ScaleData object, we can see that it's of class ScaleData, and we have a bunch of information on the number of genes, the number of cells, the assays, what are the row names, what are the column names, and so on and so forth. Since this object is of class ScaleData and that is based on single cell experiment class, the way you retrieve certain information is different than how you do it in Seurat. So I will show you how you extract certain information that you might want to take a look at. So the first thing is to get cell metadata, and for that we use a function called callData, and we provide it with the ScaleData object, and that gives you the cell metadata which has information about the cells. So here you can see we have all the information that we had in the Seurat's object metadata, which involved the information about the cell belonging to what population, what cluster, as well as the number of counts and the number of features. The next thing you would want is to get feature metadata or gene metadata. So for that we use fdata, and we type in, or we provide it the ScaleData object, and here you can see that it is a data frame, but it only has these many rows but no columns. If you take a look at the row names of this gene metadata, you'll be able to see that the row names are the genes here. So essentially it requires a column called gene short name. So we can add that column here by creating a new column called gene short name and assigning it with the row names of the feature data. And now when you do fdata CDS, you'll be able to see that you have the row names as the genes, but you also have another column called gene short name with the same names, that is the gene names. And lastly, if you want to get counts, you can simply type counts and the name of the object. So it will provide you with a sparse matrix of genes as rows, the columns as cell IDs, and the numbers as the counts. So we have converted the Seurat object to an object of ScaleData class. We also learned how to retrieve information from ScaleData object. To build a trajectory, I want Monocle3 to use the clustering I previously performed in Seurat. So for that, I need to retrieve clustering information and store it inside appropriate locations in the ScaleData object. Monocle3 also has functions to cluster cells where it not only determines the clusters but also partitions. And these partitions are nothing but these are superclusters. So essentially I need to change three things. I need to add the partition information, I need to add the clustering information from Seurat, and also UMAP cell embeddings, which are UMAP coordinates. So let us start by first assigning the partitions. And before I do that, I want to show you where the cluster information is stored inside the CDS object. So CDS object also has a lot of slots like Seurat, but the slots are different, of course. So there is a cluster slot within the ScaleData object, and within which you will find your UMAP and all your clustering information inside the UMAP, which has partitions and clusters. So essentially what we want is we want to assign all the cells to one partition. And the way we do that is we need to create a named vector or a named list. And for the named list, the names should be the cell IDs and the values should be the partition values, which is 1, because I want to assign all the cells to one partition. So to create a named list, we start by creating a vector of the values. So for all the row names, that is the cells in the call data, we assign it a value of 1. So we repeat it the value of 1 for that many times, that is the length of the number of rows, or that is the total number of cells, and we assign it to a variable. And this should be a vector. So when we look at this, we see 1 being assigned that many times, that is the total number of cells. And for the names for these values, I want it to be the names of the cell IDs, that is all the rows in ScaleData. And then I want to convert this as factor. So taking a look at this is exactly what we wanted. So this is a named list where the names are all the cells and the values are all ones. So we can use this named list and assign it to the partition. So the partition information is stored in cluster slot under UMAP, and I want to assign this named list here. So when you take a look at these partitions, now you will see that then you'll see the values, the named list that we just created. So we can see that all the cells are being assigned to one partition, that is partition 1. Next, we assign the cluster information, and we can get the cluster information from our Seurat object, that is the active ident. And when you run this, you can see which cell belongs to which cluster. So we can store this inside a variable which is called list cluster, and let's assign this list cluster to the slot within the UMAP clusters in the cell dataset object. So now we have saved the information, clustering information from the Seurat object inside the cluster slot in the cell dataset object. And lastly, we need to save the UMAP coordinates from the Seurat object inside the CDS object, that is cell dataset object. And I want to show you where these embeddings are saved inside the cell dataset object. So when you just type in CDS and run, in addition to various types of information, you'll also see a slot that says reduce dim names, and there are 2 dimensionality reduction methods, and these are the places where actually these cell embeddings are stored. So basically we need to store our UMAP embeddings from the Seurat at this location. So to access this location, we will go into this slot which is stored inside the int called data inside the list data, and within the list data you will find reduced dims, and within reduced dims you will see embeddings for PCA and UMAP. So we need to change the embeddings for UMAP and assign these embeddings and set these embeddings to the Seurat UMAP embeddings basically. So we can get that by going to our Seurat object and going to reductions, and within reductions we'll find UMAP, and within UMAP we will find cell embeddings. So essentially these are our UMAP cell embeddings in Seurat, and we need to assign these values to the UMAP, and we need to store basically these values inside the UMAP slot in the CDS. After having made these 3 changes, that is assigning all my cells to one partition, saving the cluster information from Seurat, and also saving the UMAP embeddings from Seurat inside the cell dataset object, now I want to visualize my cells, and I want to visualize it 2 ways. I want to view all my clusters, as well as I want to also view the cluster annotations. So let us start by plotting the cells, and plot cells is a function in Monocle that allows you to generate it's equivalent of dim plot. So it allows you to generate UMAP plot and visualize your cells. So the first parameter is going to be the cell dataset object. Followed by that, we want to color cells by cluster. We do not want to label groups by cluster, because we want to place only one label at the center of all the cells that is carrying that label. We want to increase the label size. So group label size, I'm setting it to 5. And I also want a legend, so I'm going to place it in the right, and we are going to assign this to a variable called cluster before trajectory. Since we haven't learned the trajectory yet, we haven't called the function to learn the trajectory. We are just visualizing our data. Now I'm going to create another plot, and this time I want to color my cells by redefine cluster. That is the cluster annotation. Again, I just want to place one label for all the cells that is carrying that label instead of placing multiple labels. So I'm going to set the label groups by cluster as false. I want to set the group label size to 5, and I'm going to set some colors for each annotation. And I need a legend as well, so I'm going to set the legend position to right. And now I'm going to assign this to cluster names, a variable called cluster names, and I'm going to run this. And now let's visualize these plots side by side. So cluster before trajectory and cluster names. So these plots show that the clustering information that we transferred from the Seurat was used to build these clustering plots, and we retain the clustering. So in Seurat as well, we identify 9 clusters, and these are the exact same 9 clusters with the exact same topology. So we would want to go ahead with this and generate a trajectory for these cells. So for the next step, we would want to actually build a trajectory now. So we call the function learn graph, and we provide it with the cell dataset object. And for use partition, we will set this to false. If this is set to true, then it will use partitions and learn a disjoint trajectory for each partition. But since we know that all our cells follow one trajectory, we will set this to false, and we reassign it back to the cell dataset object. Now that this is finished running, let us visualize the trajectory. So we can use the plot cell function again, provide it with the cell dataset object, and this time color it by the defined cluster. Label groups per cluster as false. We don't want to label branch points. I don't want to label the roots, as well as do not want to label the leaves. And the label size could be 5. And now let's run this. Looking at the plot here, we can see that a trajectory has been learnt, which is going from one end of the cells to the other. And also all of the cells are connected by one trajectory. We do not see any disjointed trajectories. In addition to that, we also see some branch points here, and a possible explanation could be that when we were grouping our cells, when we were clustering, we could not distinctly separate out all the cell types into separate clusters, and hence we could be seeing these branch points because there could be cells from the later stage or the earlier stage being present, and hence the branching is present in the trajectory. But this would make more sense once we know which cells are at the beginning of the trajectory and which are at the later stage by ordering these cells in pseudotime. So the next step would be to order the cells in pseudotime. So if you recall from the presentation, I mentioned that cells which are in the earlier state would have smaller pseudotime, and cells which are at the later state will have higher pseudotime. So let us go back to the RStudio. And to order cells in pseudotime, there is a function called order cells. We provide it with the cell dataset object. We mentioned the reduction method, and that is UMAP. And the third parameter is root cells. So we need to tell which are the cells which are present at the beginning of the trajectory, and this is based on the prior knowledge. And since we have all the B cells here, we know that the start point for these cells would be the pro B cells. So we need to give the root cells the parameters of all the cell IDs that are pro B cells. So the pro B cells belong to cluster number 5. So fetching that information, or rather the cell IDs corresponding to cluster number 5, and providing it to the root cells. And once we have that, we assign it back to the CDS object and we run this. Now that this is finished running, let's plot our cells again, and now let's visualize our cells based on pseudotime. So now I want to color my cells according to pseudotime, and the rest of the parameters would remain the same. So label branch points false, label roots false, and label leaves false. And now let's run this. Zooming in on the plot here, we can see that the probe cells which we provided to the monocle as root cells have the lowest pseudotime, and as cell progresses along the trajectory, they have higher and higher pseudotime, with these set of cells having the highest pseudotimes. So this indicates that this is the start of the trajectory, this is the starting state, and this is the end state in this trajectory. Now let us visualize what is the range of pseudotime for each type of cell. So you can get pseudotime values using the pseudotime function, and it gives a pseudotime value for each cell. So let's store this information in CDS in a variable called Monocle3 pseudotime, and let's save these values. And you can find these values in CallData, which is the cell metadata. So you can see there is a new column being created called Monocle3 pseudotime, which has all the pseudotime values. Now let us save this into a data frame. So let's convert this and save this into a data frame called data.pseudo. And when you click on data.pseudo, this is the entire call data along with the pseudotime values for each cell type. So let us use this data frame to create a ggplot. So for the x-axis, I want the pseudotime. For y-axis, I want redefined cluster. I want to color it by redefined cluster. I want it to be a box plot. And when you run this, you will be able to see the range of pseudotimes for each cell type. So let us reorder this plot. So we'll reorder the y-axis according to the median values of the pseudotime. So let's do that and rerun this. And zooming in on the plot, we can see that the cell types are arranged according to the median values of the pseudotime, and we can see that the pro-B cells have the lowest pseudotime and the plasma cells have the highest median pseudotime. So this gives us a sense of trajectory as to what are the various stages that B cell go, right from the pro-B cells to cycling pre-B to pre-B regulatory B cells to immature B cells to naive B cells, memory B cells, and finally ending up in plasma cells. And this trajectory or this lineage also corroborates with what we found in the literature that was experimentally validated. So this trajectory analysis is now helpful to look at genes that change their expression going from right from the pro-B cells and until differentiating and ultimately becoming the plasma B cells. So it would be interesting to know which genes are differentially regulated or activated or repressed during the earlier or the later stages of this trajectory. So to find genes that change expression as cells progress along a trajectory, we will basically be using a function called graph test that tests the genes for differential expression based on the low dimension embeddings. So basically it uses a test statistics that tells whether the cells at nearby positions on a trajectory will have similar or dissimilar expression levels for the gene that is being tested. So let's run the graph test function. Test, and the first parameter is cell dataset object. For neighbor graph, we provide principal graph and we provide 4 cores. So this is exactly the way that they have provided it in their tutorial, so I'm just following the exact command, and the output will be differentially expressed genes for B cells. And let's run this. Once this is finished running, let's take a look at the output data frame, and here you can see that It has a bunch of information including the p-values, the q-values, and the statistic values. So essentially we would want to get only those genes that are significantly differentially expressed, and we can filter that using the q-values. So in addition to that, I would also want to visualize the highly significant genes as a feature plot in the Seurat data. So let us do that. So we get the output data frame, and let's arrange by q values and filter for those genes where the status is okay, and just get the top few genes. So apparently I've not spelled the q values correctly. It is q value, and run this. So essentially these are the top genes that are differentially expressed. So let us create a feature plot and use the Seurat object, and for features let's plot the first three genes, which is E2F2, STMN1, and CD52, and let's run this. Zooming in on the plot, we can see that there are certain genes that have high expression when cells are in the earlier stages, and there are some genes that have high expression when the cells are in the intermediate or more differentiated state. So understanding and looking at these differential expression genes can give us an idea on the pathways or regulatory mechanisms that are differentially regulated when the cell courses along a trajectory. So these differentially expressed genes can be leveraged to perform pathway analysis or gene set enrichment analysis to understand what pathways are differentially expressed, especially when one is trying to study the mechanism of a response to a certain therapy, or maybe understanding the mechanism of development of resistance to a therapy. Lastly, let us visualize the pseudotime values in Seurat as a feature plot. So let us add the pseudotime values to a Seurat object. And let us create a feature plot now. And let's run this. So this is the pseudotime values, and now we are visualizing it in Seurat, where the cells with the lowest pseudotime are in gray, and as and when the pseudotime increases, the intensity of the color goes on increasing. Let us change the idents of the Seurat object to have the cluster names, and let us recreate the feature plot with the labels. So now we can see that in addition to just the pseudotime and clusters, we also have the cluster label, which helps us understand which cells are having the lowest pseudotime and which are the cells having the highest pseudotime. So that brings me to the end of this video. I hope you found this video helpful and informative. As always, I will upload my script to the GitHub and will add the link to the GitHub in my description section below. So if you found this video informative, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.